poliovirus. Okay. So this is the classification of the picornability. That is it contains several genera, Intero, Rhino, Hepato, are the human pathogens. Intero and Hepato are important because they infect via gut, resistant to acidic pH of our gastric juice, and the proteolytic enzymes and bile salts. Okay. So that's why they are they infect us via the GI route. Okay, and rhinovirus that cause the common cold, they multiply in the lungs. Okay, and in today we will be discussing on the poliovirus. Okay, the poliovirus 1 to 3 positive virus group: ecovirus, enterovirus, rhinovirus, hepatovirus, the hepatitis A virus. These are the viruses which comes under the family Picornaviridae. Okay, and we will be focusing on the poliovirus. And these viruses are inactivated when heated at 55 degrees centigrade for 30 minutes, and magnesium prevents inactivations. Okay, and since they are the viruses, they cannot be cultivated in artificial culture media, and they require different cell lines, okay, cell culture system to cultivate the viruses. That's why they can be cultivated in the cell line cultures derived from the human or from monkey kidney, testes, and vessels. Some background noise is there. Please make your microphone.
यति सागले के भयो बिना काट्दा पनि छ
Okay, and this group of virus requires the specific memory receptor for the infection. Now, let us discuss regarding the properties of these viruses. They are the naked virus with the icosahedral nucleocapsid symmetry, and the size is 30 nanometer. Okay, they are very small. The outer capsid layer contains 60 proteins developed by the protomers, and these protomers or these protein subunits have 4 peptides, that is the BP1, BP2, BP3, and BP4. Okay, and these are important in pathogenesis. These are the structural components which are important in pathogenesis as well. Okay, and they have the linear molecule of single stranded RNA, positive chain RNA with 7 to 8 kilo base pairs in sequence. Okay, they are called as picoNAb because their RNA is very small. See here, only the 7 to 8 kilo base pairs. Pico means very small, RNA means ribonucleic acid. Since they are the RNA virus, which contain the positive sense single stranded RNA with very small genome size, only 7 to 8 kilo base pairs. Okay, so this is the genomic structure that contains the structural regions that code for these viral proteins BP1 to 3 and 4. Okay, and some non-structural regions that code for some regulatory enzymes and all those things. Okay, so now this is the electron micrograph of poliovirus. It's the real image taken in the electron microscope. So these are the polioviruses. Okay, that is being described here. So these viruses attach with the specific receptors, penetrate inside the host cells, Uncoating takes place and synthesize the viral components, and finally these viral components or the precursors assemble and then finally got released from the cell. So this is the replications, how they replicate. Okay, the difference see here, the receptor is very important to engage. Since the receptor for the poliovirus are here in the GI, gastrointestinal tract, that's why we acquire infection by ingation. Okay. See, this is the outline how these viruses replicates. First, they interact with the receptors, enter inside on course, and there will be the replications. In replication process, here first there will be the function of the genome copies, as well as there will be the transcription and translation for the production of structural components. Okay, and once the genome copy and the structural components synthesized and they will be further, and finally they will be out from the host cells. So this is the outline how these viruses replicates within the cells. See here, the specific receptors that is the VPR receptor on the cell membrane of the host cells. Okay, we should remember this one, VPR receptor, viral specific. And they enter inside via the process of endocytosis, or simply we can call as the receptor mediated endocytosis, also called as pyrodesis, and then they enter into the cytoplasm, and the outer capsid layer release out or the broken out, and the newly exit is called as uncoating. Okay, and this is uncoating, and then after uncoating, two process takes place. One is the genome replications, okay, which is required for the newer progeny, and another is the transcription and translation. Okay. Transcription means the information that is transcribed into the genome copy, and that will be expressed by the translation process, and the structural and the regulatory proteins will be synthesized here, and finally they got accepted. Okay. This is the replication of the poliovirus. Okay. Now, the structure and the replication of this much for the structure and replication of the poliovirus. So now the important one is the pathogenesis, which can be asked even in the examination also. Okay. As I have already mentioned, the receptors to enter inside the host cells is present in our GI. That's why the mode of infection in this case, or in poliovirus, is the fecal route. That means the poliovirus which are there in the GI, sorry, in the fecal material, contaminated fecal material, if we ingest them, then we acquire infections. And how it progresses. First, what happens? They multiply locally in the intestine and the oropharynx. They multiply locally, and the antibodies found in blood are present with abortive and non-paralytic poliomyelitis. Means once they multiply in the GI, it doesn't mean they results in the clinical conditions in the poliomyelitis. The patient can be asymptomatic, but in those conditions, if we try to find out the antibody against the poliovirus, then we may found. Okay, I'll try to explain it clearly. It is very clear what happens in this. Okay, this will be very much clear to you. See what happens. First, once you engage the organisms, they replicate locally in the oropharynx and the intestinal mucosa. That is called as alimentary feed. Okay? And then during these multiplications, these virus split the tonsil and multiply their patches, cervical and mesenteric lymph nodes. Means the local tonsillar lymph nodes or the malt which are present in the GI, okay, or the lymph nodes, okay, that are present in the adjacent area of the GI. Okay, there the viruses multiply. That is called as lymphatic phase. Okay, and when after some episodes of multiplication in the lymph nodes, local lymph nodes in the GI, okay, they finally enters into the blood streams. That is called as viremic phase. Okay, that is called as viremic phase. Till now, the patient will not have any kind of symptom. Even in the viremic phase, in most of the cases, almost 95% of the cases, the virus do not further spread, do not further multiply, and the patient might have some asymptomatic or non-specific features. Okay, here we can see the patient will have infections, virus will present in the blood, but will not have any typical features. Okay, some non-specific features or some undifferentiated clinical illness can be seen, but that automatically cleared. Okay, in most of the cases. But in some cases, which is very much important aspects, is in some cases these viruses via the spinal, via the nerves, they uh, 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 travel towards the central nervous system via the retrograde axonal transport. Okay, and then they may lead to the paralytic conditions. And this one is important. That is called as neurological phase. Okay, that means they will ingest, they will multiply locally, and again they multiply in the local lymph nodes, lymphatic phase, and then they intercept to the blood stream, that is by the almost more than 98% of the viremic phase do not spread further, and during that time the patient might have some asymptomatic features, and finally the patient got result. But in this case here, the viruses manage to bind to the nerve endings because they have the receptors to bind on the nerve endings, and via the retrograde axonal transport, they reach to the CNS and for the paralysis. Okay, so that is the pathogenesis of the viruses. Okay, see here that the polio virus, they can spread along the external nerves to the CNS. Okay, and along the lower motor neurons. Means these area got involved, and then patient will have the such kind of complications. That is the okay. So now clinical findings. See here, this in apparent infection without symptoms means patient will have the infection but not have any kind of symptoms. That means in the viremic phase, they do not further multiply and the patient, the infection subsided. Okay, or sometimes the patient might have mild illness also. And these two clinical findings are not important. The important one is this, because these these may prognosis to these conditions. Okay? That is aseptic meningitis and the paralytic poliomyelitis. Aseptic meningitis means they multiply in the veins and they inflame the meninges. Why they are called as aseptic? Because the viruses are not easily identified in the CSF sample. They are not cultivable because they are not easily cultivable. That's why they are called as aseptic meningitis, because it may lead to the meningitis also. And finally, the paralysis of the affected organ. Okay, so that is the poliomyelitis. So these are the stages that we see in the polio. And these two can be fatal. Aseptic meningitis, and in case of paralytic poliomyelitis, it may fatal, or the remnants, that is the disability may remains for prolonged period or till whole life. Okay, so that's why this is important. Okay. Because the patient may remain symptomatic till the whole courses, and some may lead to these conditions, and that may persist for whole life. Okay. So now these are certain terminology. For example, abortive poliomyelitis. Here the patient. Okay. Here this is the most common form. Here the patient will have some non-specific features, and the patient will recover in a few days. So that means paralytic poliomyelitis. In the non-paralytic poliomyelitis, I have already mentioned that there will be the feature of the meningitis, still multiplication of the in the CNS involving the meninges. Okay. And another is the paralytic poliomyelitis. This is important because there will be the paralysis of the affected organ. Okay. So due to the damage of these motor neurons, okay, the limbs, most commonly the limbs will be the paralyzed, it's called flaccid paralysis. This is very much important because it will persist for life. Okay. This is very important. See here, this is shown in figure. Okay. See here, due to the polio, and there is a disability till whole life. Okay. This is more about the pathologist. So now, how we diagnose? As we have already discussed regarding the diagnostic approach. The first one is the direct observation of the organism. Still, virus is not possible, but we can do electron micrography. But not present all the time. Another is the culture, that is the recovery of the virus. This is also not common because establishing the cell culture lab not easy because it is very difficult to maintain the cell lines viable and it is very difficult to prevent contamination by the other bacteria and other things. That's why cell culture is also not a big job. In cell culture, you can see the cytopathic effect. If we could manage culture system. Okay? But culture can be done in the multi-pixel cell lines or the cell lines. That is the cell culture, okay? The recovery of the virus. Okay, some indirect evidence of the CSF analysis also help us because there will be increase in the protein content and the glucose level will not alter. Okay, and WBC count not so high, but relatively it is increased leukocytes in CSF sample with the more common one is the lymphocytosis. The lymphocyte will be high. Since this is the viral infection, in viral infections, the lymphocyte will be okay. This is the indirect evidence. Okay. And now, the most important one is the antibody detection. That is the antibody against the antigens, the C antigen and the D antigen of the polio virus. If we could detect in the patient serum, basically in the fluid serum, then if there is an increase in the titer of the antibody, then in those conditions we can say the patient is having polio. Okay. So now epidemiology, it occurs worldwide. Okay, occurs in all age group, but children are more commonly affected or susceptible. Adults have gained the acquired immunity, that's why less common in adult. Okay, spread by fecal-oral route, as I've already mentioned. And the important point is this: they can excrete for several weeks in the feces, even in the after the convalescent phase or the recovery phase. Okay? The infected portions are the main source of infections because they can be spread out via the fecal material for several weeks from the infected portion. Okay, so that's why poor hygiene, overcrowding, lack of diagnosis also facilitate the spread of this disease further. Okay, and some explosive outbreaks can occur due to contamination of water supplies by sewage. Okay? So that means the areas where there is the improper water sanitation, sewage disposal systems, then in those conditions, there is the chances of having outbreak also. Okay. So now, but luckily, some preventive measures are there. That is the immune prophylaxis are there in the polio. As you all know, that is the polio thopa, that is the oral polio, and then that is the IV. Okay, that is the injectable polio. Okay, IPV, OPV, and IPV. Live vaccines, that is the oral polio vaccines, which was first introduced by the Savin, which is available, and they multiply, infects, and then uh, produce the antibody against the polio virus. These live oral polio vaccines is useful to maintain the local immunity because the antibody IgA, since we engage them via the oral route, the viruses will have the similar course as in live polio virus, okay? Natural polio virus. In similar way, the viruses enters, multiplies, but they are not, their pathogenesis has been lost. Okay. So here what happens? These live polio virus, when we ingest as a vaccine, okay, then they will provide us the local immunity. Means the antibody against this virus will be there in our GI. Once the patient will ingest, then this antibody will not allow them to enter inside the GI and enters into the blood circulation. In this way, they will be helpful. And mass oral polio vaccinations also helps in the development of the herd immunity also. So since these are the live vaccines, these also might have certain kind of complications in immunosuppressed individuals. Okay, and then in those conditions, the killed vaccines has been used, which are the injectable vaccines. And these injectable vaccines, these vaccines are relatively safer, but it requires the continuous booster doses to maintain the immunity. So this is the preventive measures. Since these vaccines are the polio is the vaccine preventable disease, through the effective vaccine policies by the government, Nepal also has announced the eradication of the polio. There are several countries that have already been announced to be eradicated or the polio has been eliminated. But till now, WHO has not announced that polio has been eliminated from the world. Okay? So though some countries have already announced as a polio-free countries. Okay? So this is more about the polio. Now, in polio, there are certain points that you must know. One is the preventive measures. Okay? It can be asked in community medicine as well as in microbiology. In microbiology, one is the laboratory diagnosis. In laboratory diagnosis, serology. Okay, serology, it helps to diagnose. One is the culture, and another one I already mentioned here, the PCR. Okay? That is the gene detection. We can also have to diagnose the case. Okay, but live microscopy, culture, okay, these are not possible in our setup. Okay. So there's a lab diagnosis part. And another one is the pathogenesis. This one is also important. Okay, pathogenesis. Right? So if pathogenesis are asked in polio, then you can write this one. This is very much informative to describe the pathogenesis of the polio virus. Okay. This was for today. If you have any questions, then let us discuss. Otherwise, finish our class.